Hello everybody, today we would like to present the comparison of soil quality in tropical rainforest affecting the growth of trangensis. First, let's meet my team.
Hello, my name is Sudipali from.
Hello, my name is Pagangit Thonabhishatkun.
Hello, my name is Pimploy Thong O. In first part, introduction, review of literature. Soil is a natural material that is formed by many things coming together, such as water, air, mineral, rocks, and etc., which arising from the decay of rock and minerals, as well as the decomposition of the remains of plants and animals mixed together. These factors come together to form soil. Soil and resources are nature that is important to living organisms because it is the source of the four necessities for living, namely food, clothing, housing, and medicine. These things can be obtained from the soil both directly and indirectly. It is also important for plant growth because it is the place where the roots cling to the stem to stand firmly and strong. It is also a source of water and nutrient that plants need to grow in each different soil condition, causing plants to receive different nutrients, resulting in different growth in each area. That factor affecting the soil physical and chemical characteristics are different. It's often caused by many reasons such as geographical environment, biological factor, earthworm will help make the soil loose, which is maybe a climatic factor such as rainfall, temperature, wise, and these factors are related to plant growth.
Ground orchids are new plants that were first discovered in Trang province. Spatogotrid is a genus of plant from the orchid family, covering relatives close to Acanthaphippium. The species discovered in Trang province is named One Pendidemon trangensis, botanic name Nelvilla trangensis. Family orchids. A new type of orchid discovered this time is a ground orchid with an underground head. The leaves are light green to dark green, raised slightly above the ground, and is less common due to environmental changes that affect the soil is making the growth factors of orchids difficult to find. Therefore, the organizing team is interested in the studying and comparing the soil quality in the rainforest of Tunkai Botanic Garden that affect the growth of Novella trangensis.
Research question: Do mineral and soil physical characteristics affect the growth of Nebelia trangensis in the humid rainforest? These are achievements that we use. Methods of conducting research. Part 1. Soil sampling in all three sources. 1. Exposure area with three different spots of nebulous trangenesis. 2. Measure the temperature of soil in the area with trangenesis 5 minutes. Repeat 3 times in all three soil sources. 3. Use small shovels to dig through soil from all three sources. Is digging 50 grams. Push one bigger. Pour salt into the bigger. Pass through, making a soil solution. 1. Pour distill water into 3 beaker containing 150 milliliters of soy. 2. Use grass loaf to stir the soy until the soy dissolves completely. 3. Leave the soy solution until the section. 4. Take the filter paper and funnel off filter the soy solution. Form soy in the beaker into Helen measure from 5. Where the substance resulting of the filtration are obtained. Take them to test the soil quality with a soil test kit. Part 3. Soil quality and inspection. 3.1. Test of nitrogens. 1. Put the soil solution of the beaker in the test tube. 10 milliliters is 3 tube. 2. Put the nitrogen test kit in 3 test tube. Check the tube as switch off. Take result for 5 minutes. 3. Compare the result with the temperature to see the amount of soil nitrogen.
3.2 Test for phosphorus. 1. Put the soil solution from the beaker into a test tube 10 milliliters each 3 tubes. 2. Put the phosphorus test kit into all 3 tubes. Check the test tube and wait for the test result for 5 minutes. 3. Compare the test result with the color template to see the amount of phosphorus in the soils. 3.3 Test for potassium. 1. Put the soil solution from the beaker into a test tube 10 milliliters each 3 tubes. 2. Put the potassium test kit into all 3 tubes. Check the test tube and wait for the test result for 5 minutes. 3. Compare the test result with the color template to see the amount of soil potassium. 3.4 pH test. Prepare the solution that want to test put in a suitable container. 2. Dip the pH test strip into the liquid you want to measure for about 2 to 3 seconds. 3. Remove the test paper from the container and wait 10 seconds for the paper to change color. 4. Compare the color on the paper with the scale on the box. 5. Check pH.
Analysis and conclusion of research. The way to find the average for is by taking the sum of all observations and then dividing it by the total number of observations. Results. Soil minerals. Form diagram 1. The measurement of nitrogen in the 3 soil sources. Show the first source had an average nitrogen of 2.33. The second source had an average nitrogen of 2.67, and the third source had an average nitrogen of 2.67. From diagram 2, the measurement of phosphorus in the 3 soil sources. Show the first source had an average phosphorus of 3.67. The second source had an average phosphorus of 4. and the third source had an average phosphorus of 3.33.
From diagram 3, the measurements of potassium in the tree soil show that field 1 has two average potassium value. Soil 2 has average potassium value of 3.33, and field 3 has an average potassium value of 1.67. From table 4, the comparison of three soil pH value is what found. This soil once have an average pH of 6. Soil 2 has an average pH of 5, and soil 3 has an average pH of 6.33.
From table 5, the comparison of the soil temperatures of the three sources, it was found that the first soil had an average temperature of 28.33 degrees Celsius. The second soil resource had an average temperature of 28 degrees Celsius. The third soil source had an average temperature of 28.83 degrees Celsius.
Soil moisture. From table 6 showing the moisture contents of the three soil sources, it was found that the first source has an average moisture content of 3.12. The second source had an average moisture content of 3.2, and the third source had an average moisture content of 3.8. Next, discussion of results. The study of comparing the soil quality in area that affects the growth and development of Nevela trangensis in Thung Khai subdistrict, Yan Ta Khao district, Trang province, Thailand. It was found that the average nitrogen content in source 1, 2, and 3 was 2.33, 3.33 and 2.67 respectively. The phosphorus content was 3.67, 4, and 3.33 in source 1, 2, and 3 respectively. The potassium content was 1.33 and 1.67 in area 1, 2, and 3 respectively. The pH value were 5, 6, 5, and 6.33 in source 1, 2, and 3 respectively. The temperature was 28.33, 38 and 38.83 in source 1, 2, and 3 respectively. The humidity was 3.12, 3.2, and 3.8 in source 1, 2, and 3 respectively.
Finally, conclusion. From the study of comparing the soil quality of three sources for the growth of Nephila trangensis, it was found that in the first source and the second source, nitrogen values were equal and higher than the third source, while phosphorus in the second source has the highest value, followed by the first source and the third source in that order. Potassium in the first source has the highest value, followed by the third source and the second source in that order. pH in the third sort has the highest value, followed by the first sort and the second sort in that order. The temperature in the third sort has the highest value, followed by the first sort and the second sort in that order. Moisture in the third sort has the highest value, followed by the second sort and the first sort in that order. It can be seen that in the first sort, nitrogen and potassium are higher. In the second sort, nitrogen and phosphorus are the highest, and in the third sort, temperature and moisture are the highest. Additionally, it can be observed that the soil in the third sort has the highest value of alkalinity. Thank you for
Thank you for
attention.
attention.